Thanks so much for having me. Really appreciate it. My name is Frank Piccininni from Spadefoot Design and Construction. I'm here. Three important points that I wanted to kind of really get off the bat here. The first one is that I don't speak for SUNY ESF. I'm here on, I cleared the talk and the slides with the New York State chapter of the American Chestnut Foundation. But given the sensitivity of the topic here and what's all happened lately with the national, I just wanted to make it clear that I'm not here speaking on behalf of ESF. The other thing I wanted to make clear is that I'm an ecologist and a lawyer, but not a geneticist, nor am I an expert in the deregulation process. So I'm somewhat in learner mode when it comes to those parts of the American chestnut, deregulation and whatnot. So if there's questions involving some complicated genetics, I might have to scratch my head and say, I don't know, I'll try to find out. And the third thing is, I realize that I'm the last guy here between you and lunch, so I'll try to be very quick on this one. So keeping up with the Castanillas. Yeah, so just briefly, and I know this is a conservation update, and this has been talked about before, but what's the big deal with the chestnut? So within their range and within their niche, really mid-slope in upland forests, these species were absolutely dominant. They were important wildlife food. I'm just trying to see if I don't have my glasses on, so I can't really read my notes here. Ecologically important species. So just both for wildlife, for Native Americans, and for other settlers, the nuts were a really important part of the economy. And unfortunately, they were decimated by Chinese chestnut blight. Must have been watching a train wreck in slow motion. I couldn't imagine, witnessing this. Although we're getting a taste of it, we'll hear from Mina Versera afterwards about the beech leaf disease, something similar. But what happens is that this foreign pathogen was introduced, thought to be by the horticultural trade. People brought in Chinese chestnuts because they were pretty, and what happens to the chestnut trees, unfortunately, is that it basically girdles the tree. So on the xylem and the phloem, the circulatory system of a tree is in the top layer. And what happens is that the blight sets in and really cuts off the circulation of a tree. And the trees the stems, the above ground growth kind of just falls over and dies, unfortunately. And we were subject to what was referred to as salvage logging. So when the blight was happening, a lot of folks figured, oh, well, they're all going to die anyway. Let's cut them all down and actually put the logs to economic use. So we were robbed of an opportunity to see if there's any really natural resistance, which is really sad. Nevertheless, the thought is that it may not have come to fruition because the blight is particularly devastating. And so now they're functionally extinct. They're actually all over Long Island, believe it or not. So what happens is that the root stock remains, the above ground stem dies, falls over every 30 years or so, and a new shoot will come up and the process starts anew. So they're relegated to the understory. They're kind of like big shrubs with ugly bark, and that's what they are now. Once former great. What are we doing here locally with the chestnuts? So when I was with Save the Great South Bay, we started an awesome iNaturalist search, the search for the American chestnut, and it's populated. There's a lot of American chestnut left here on Long Island, which is awesome. My co-director of the New York State chapter, Long Island co-director, Nico, he's actually out there hand pollinating these species. So he's climbing finding these vestigial populations, taking pollen, and just literally with a toothbrush pollinating these flowers, and then going back collecting the nuts. They're being propagated by Nico and Long Island natives, which is awesome. So we have Long Island ecotype, Long Island ecotype genetic American chestnuts that we're putting in mother orchards all across Long Island. And what's the point of that? The mother orchards will, once the transgenic stock is made available and deregulated, we're going to crossbreed the Long Island genetic chestnuts with the blight resistance transgenic chestnuts. So what you'll have is Long Island, at least 50% Long Island ecotype with some blight resistance, which is awesome. We're doing a little bit of mud packing. We're actually taking mud and kind of, you basically press it against the bark and all these cankers, and it's been shown to extend the life of the individual stems. And we have some, it's really in its nascency, but we have some planned research on figuring out the niche selection of American chestnuts. So when we do have blight-resistant stock to revegetate, where are we going to put them and how are we going to put them there? So it's sort of complicated because if you look up online blight-resistant chestnuts, you get all kinds of stuff out there. There's the long-standing practice was the 10 minutes left. Is that what that means? Okay. Was the back-cross hybrid practice, and so what the American Chestnut Foundation was trying to do for the longest time was to introduce Chinese chestnut genes into by crossbreeding it with the American chestnuts. And the hope was to develop a hybrid that had the blight resistance that the Chinese chestnut has with American chestnut genetics. But turns out it's more complicated than we thought. The control for blight resistance is controlled on apparently a lot of different genes. And so, unfortunately, with the American Chestnut Foundation, the back-cross hybrids that exhibit blight tolerance are predominantly Chinese chestnuts. So I personally, as an ecologist and as a restoration ecologist, I don't find the back-cross hybrid program very promising anymore. Fortunately, the SUNY School of ESF has worked in a transgenic lab. Basically what they did, this is pretty phenomenal to me and a little bit above my head to be honest, but they took a wheat gene and inserted it into the genome of the American chestnut and it confers blight resistance to it. So there's 3 different lines that they have right now: Darling 54, Darling 58, and Darwin. I'll spare you the talking about the different ones, but right now there's a petition for deregulation. And so any GMO plant species, when it's being submitted to the FDA and the EPA, you have to kind of make sure that the plant is not going to just, a mutant wheat, let's say, is not going to just take over the environment. And so there's a whole process that these GMO producers have to go through to get to deregulation status. And once you're with you get deregulation status, you can actually put the stuff out there without fear of spreading, or at least according to the government. Unfortunately, the project proprietor, the principal researcher, Dr. Bill Powell, recently passed away of cancer, way too young. So that was a big blow to the American Chestnut Foundation. And this school of ESF has experienced some adversity, let's just say, around this program because they recently found out that the Darling 54, which is an older line, and the Darling 58, which is the one that they were looking to get deregulated, there was just a mistake in labeling. And so all the studies were still good. It was just the name of the line that was made a mistake. So upon figuring this out, ESF went to the regulators, updated and amended their petitions pretty quickly. I have a note here about chestnut business, right? This one's sensitive, and I contemplated taking the bullet point out. But ESF, it recently came out to all the chestnut nerds that are out there that ESF was not going to grant an exclusive license for distribution on any of the transgenic lines to any individual organization. And I'll leave that right there, but contemporaneous with that coming out, unfortunately, the American Chestnut Foundation has pulled their support for the deregulation petition and basically went on the offensive and saying, we no longer support the work of SUNY ESF, which is tough. It was a surprise to ESF. They're reeling from the loss of the project leader, and the National American Chestnut Foundation pulls kind of the rug out from under them. They cited in their chestnut chats red flags. So there's a couple things that they're looking at. So, and this is pretty complicated. The inducible promoter versus constitutive promoters. So what I gather from what I read, and it's about whether or not the gene for blight resistance is turned on at all times. So the Darling 54 and the Darling 58 lines, they just put the gene in and it's just on, producing this OXO enzyme, which takes away from the virulence of the blight. But it's thought that perhaps that because it's on at all times, there's some effect on the efficacy of the plants. Like, do they grow slower because the gene is on at all time? So they were citing the fact that the Darling lines, Darling 58 and 54, are using constitutive promoters as opposed to inducible promoters. It's an inducible promoter, in contrast, turns on when there's like a stress inducer. So they kind of hung up on that. They use growth rates in common garden experiments. So basically, they had a bunch of wild types and a bunch of Darling 54, not 58, 54 lines. And they said, oh, it looks like the transgenic stock is not growing nearly as fast. Yellowing leaves on the transgenic stock is what they were kind of citing as a big example. There's some concern for the chestnut crops, whether or not if we had inferior material out there, whether or not that would have economic burdens to the Chinese chestnut growers and people who are spreading that. And then this wasn't in none of their chestnut chats. One of the national members told me, oh, did you know that Monsanto is involved in the research for chestnuts? And so, aren't you concerned about that? Which I'm not. I'm an ecologist. But so what was ESF's response? It was a very measured academic response, which is to say, we're looking at this, and we don't. They had some really good, interesting things, but the New York State chapter of the American Chestnut Foundation has been doing some talking on behalf of ESF. So first of all, the process of deregulation. What is the purpose of it anyway? It's to make sure that we don't have any, mutant, like I said, wheat plants, taking over the ecosystem, in the Atlantic grasslands or something, right? So it just doesn't even make sense to me personally, and I'm editorializing as an individual here, not on behalf of an organization. But if the argument is that the transgenic stock is not as effective for a restoration species, well, it doesn't really matter from a deregulation standpoint anyway, because the whole point of the deregulation is to keep our native land safe. So if you're saying that it's not going to do well for restoration, well, it doesn't really matter because it should still be deregulated, because once it's deregulated, all the other lines, like the Darwin line, which is a constitutive promoter, inducitive promoter, it's a lot shorter of a path for approval and distribution. So, this whole bit about inducible versus constitutive promoters, the Darling 58 line is the one with the constitutive promoter that's in there for deregulation. The inducible promoter, they have one already, ESF does, the Darwin line, that has this inducible stress, reaction promoter. So, the American Chestnut Foundation said, tisk, tisk, this is a bad, you're using the wrong promoters. And ESF is scratching their head saying, well, we have the other one ready to go. Once the Darling line is deregulated, then we can go out maybe within 3 months with the Darwin line. So the growth rate in common garden experiments, I actually haven't seen this data. The national claims that they have data, but forgive how blurry and terrible this picture is. It was a screen grab from the chestnut chats. But it just kind of shows you, this is the big picture that nationals banding about as far as like, oh, the wild types grow more quickly than the blight-resistant transgenic stock. And what you'll notice is that the one on the left is in the sun, whereas the one on the right is in the shade. So, I'm not saying this makes for a bad study. The study hasn't come out with this, with the pulling of the support from national. Loss of blight resistance, folks, this one's tough too because it's based on one trial, the Purdue trial, and what SUNY ESF has learned over time, and the American Chestnut Foundation, the New York chapter of the American Chestnut Foundation, is that it's really easy to get a false positive on whether or not it's OXO positive or not. There's cross-contamination of samples. So it's not even clear that the Purdue trial actually had the stock from ESF. It was actually potentially from their own lab. Yellowing leaves, I think they're thought to be overwatered, is the simple answer for that one. And concern for chestnut crops, again, this is for restoration. This isn't for food sources. And Monsanto is the boogeyman. it's about time that they're doing something good, to be honest. And so, you know, if they're out there and they're helping with research that can really help with the restoration of the ecosystem, I'm all for it. And if there's really good scientists that are volunteering their efforts for this, you know, that's fine to me. So I say here, a New York state of mind. This is a wild-type chestnut we found on one of Spadefoot's projects, our project manager, Brittany here. But I recently found out that the New York State chapter of the American Chestnut Foundation is a completely independent organization, separate bylaws and everything. And so it's named the American Chestnut Foundation, but stay tuned because we're, okay, that's enough said there. We're going to continue to support ESF and the transgenic work. The backcross hybrid, from my own personal experience, is an absolute failure. It's all Chinese chestnut. And we're looking locally to crossbreed it with other local ecotypes. And ESF is looking, it turns out the equipment that they have, and I know the stop sign came up, but the equipment that they have at SUNY ESF can be used for a ton of other trees. So they're working on American beech trees, elms, and a whole lot of other things. So thank you so much for your time. I really appreciate it.
Questions? Looks like everyone's hungry.
Yes, sounds like it. Maybe one.
Hey, John.
Excellent talk. Thank you so much. A quick question in regard to there's historic photographs from the 1800s showing these gigantic redwood-sized trees in the, I guess it's in the Carolinas and so forth, the Great Smokies of gigantic chestnut trees. Did Long Island trees that we know, did they ever grow to that size, or were they limited because we're mostly a sandbar here? And is that part of the reason why the local chapter would be pursuing the crossbreeding with local genotypes?
Yeah, no, that's a great question. So, you know, if you actually look at a lot of the chestnut literature, you know, people describe it as these big giant trees that were everywhere, but it's really a very specialized tree. It co-associates with like chestnut oaks and beeches and stuff, but it's really mid-slope on inland hills. So think about West Hills County Park as a good example, like that sort of habitat. North of the glacial moraine, there's a lot of it, but even south of the glacial moraine, there was a ton of it. And in terms of the local genetics, yeah, that's exactly why we're going out and trying to save the genetics of the Long Island chestnut so that we don't have, you know, SUNY Syracuse chestnuts all on Long Island. At least we'll have some of the local ecotype in the genome.
Thanks. Just one other quick question. Given, what's the timeline, do you think, for approval by USDA, FDA for the SUNY ESF? I know I've read some of the controversy about that.
Right.
And then, you know, we're looking at the next year for approval, perhaps. And then, what is the timeline for getting a local version of that could be plantable on Long Island?
Yeah, that's a great question. So first of all, I'm like very much out of the loop with that. That's really between ESF and the regulators. And again, we're talking about government. But I've been hearing 3 months for the last year or so. So, you know, I don't know.
They've been working on it for about 6 years.
They've been working on it for a long time. And of course, this whole, you know, switched at birth labeling error stuff causes some pause. It might cause some further delay, but to my understanding, we're pretty close. I'm not sure that the Darling 54 is actually ever going to be released. I think they might be looking at the Darwin line. And in terms of like timeline getting it out here on Long Island, your guess is as good as mine. I know that we've been busting our tails to get mother orchards out here, and I know that you know, me doing talks like this and you know, stirring the pot a little bit on the internet is currying some favor with some of the folks that have control of this. So, you know, hopefully in the next couple of years, we might have some blight-resistant American chestnut to, you know, be Johnny Chestnut, see it out in our local woodlands.
Thank you.
Hope. Yeah.
One more, Steve.
Yeah, hi. Great update. Thanks a lot. I assume that deer like chestnuts a lot, and you know, eating the seedlings. Could you ever restore them without controlling the herd?
Yeah. No, Deer is a tough issue, and as you know, the owner of a company that does ecological restoration, that's our entire business, it is an absolute struggle. And so in deer lands, when we have these chestnut, plantings, we have to use deer fence. Typically we use like the poly fencing with tree stakes has been what we've been generally using. But until they get above the browse line, they're absolutely open for herbivory. And even after that, every fall, the bucks are coming around and, knocking around our whips. So deer, can you restore the chestnut without deer? I think that, can you restore the chestnut with so many deer? I think it's gonna be more expensive because we have to figure out how to protect the plantings from the deer. And it's a really troubling thing, and I don't want to get in trouble with PETA, but there's some management that is absolutely necessary, and I often hear you saying that. Yeah.
Okay. Thanks, Frank.
Sure.